Then we start. So hello, my name is Miriam Löffler from Gau Löwe, and it's my pleasure to welcome you in the name of the EFFoST Health and Food Working Group to our first webinar in 2026. And I'm already excited about today's program. For those who do not know us yet, we are the Health and Food Working Group with international members from academia and industry. And we share both expertise and passion for the related research and also the topics you're going to see today. And as a working group, we aim to tackle health and nutritional challenges by focusing on the development and implementation of technologies and the formulation of innovative future food products. The group will function as a connecting link between future trends in food science and the evolving dietary requirements of consumers, bridging together experts and stakeholders from health, nutrition, and food science and technology. And more about us and what we do can be found on the EFFoST homepage. It would have just been a bit too much for today. All right, let's move on to the exciting program of today. And already a big thank you to our keynote speaker, Oliver Schlüter, the PhD students who will present their work, and also to you, the audience. Let's start with some housekeeping rules first. The following webinar will be recorded and archived on the EFFoST webpage and YouTube channel. The record will also be shared with all participants afterwards, so you don't gonna miss anything. Very important, please turn off your microphones unless invited by the moderator. If you have questions during the presentation, please type them into the Q&A chat box, and our moderator will be tracking these for the Q&A sessions. If not all questions can be asked or input shared, we will provide it to the speakers afterwards. With this, let's take a look to the outline of the day. I have already introduced shortly the working group. We will then start with a keynote by Oliver Schlüter on shaping the future of food production and consumption together. Next, we have 3 rounds of presentations from our PhD researchers. The first round focuses on product formulations. We will introduce the speakers in a second. The second one will focus on novel food ingredients. And the third one on food processing strategies. This part of the program will be moderated by Robert Sevenik from the TU Berlin. After each of the 3 rounds, he will guide the Q&A session based on your questions and input from the chat. He will also act as a timekeeper as the presentations are scheduled for 5 minutes each, so please try to keep the time. And now, I would like to hand over to Marius Zarianos from ATB, who will introduce today's keynote speaker.
Yes, so thank you, Miriam, and it is my pleasure and my honor to introduce Dr. Oliver Schlüter, who is a senior scientist at the Leibniz Institute for Agricultural Engineering and Bioeconomy in Potsdam. Here he's a spokesperson from our program area called Healthy Foods, and he also holds an adjunct professor position at the University of Bologna. So the floor is yours.
Yeah, thank you very much, Marius, for this kind introduction. And I would like to thank all the chairs of this new working group. It is very exciting, and I'm really happy to see that you put this nice program together. And to be honest, I think it took nearly 10 years that we got this started, because the first idea was coming up, I think, 2016. And I'm really happy that 5 enthusiastic young researchers are taking over and put this thing to run anyway. So the idea today is just to share some of our views with respect to future food systems. And I would like to introduce some of our approaches with respect to future food production and selected examples. So, as you know, especially with respect to health, we have to deal still with food losses. There was a recent publication in Nature Food mentioning that food losses are still 1/3 of the overall food production. That means 1.3 billion tons a year, and this is immense, and this also causes hunger. On the other hand, in Lancet recently there was a publication that during the last decades, more or less obesity was increasing in all countries around the world. So this is another task we have to consider during our research with respect to future food systems. And beside the health aspects and the aging aspects, we have the growing population. But we have to consider as well the urbanization. That means increasing cities where transportation is more difficult from the suburban area to the city center. We have to ensure that healthy food is not a question of money, so that also people having not that much money can have food which is healthy and safe. And also digitalization is a hot topic for future food systems. We all know about the discussions of the potential of AI and so on. And last but not least, the term resilience is more and more important due to unexpected crises we had to face during the last years. And especially sustainability is a topic with respect to planetary health and One Health approach. And there was a paper also recently on the sustainable food protein supply, together with some colleagues from the Leibniz Association, where I'm also from. Our institution is part of the Leibniz Association. And then some years later, it was clear that probably sustainability is not enough. So there was another position paper on sustainable and resilient agri-food systems. And here sustainability is linked to resilience. And in this paper, it was also stated that to overcome the challenges, taking advantage of alternative food sources is important, and also innovations with respect to technologies. So, and regarding the food sources, we have a lot of new food sources, maybe not that new, but for, let's say, for our region or in Europe, it is new, like fungal protein, microalgae, macroalgae, cultured meat, and insects. But mainly we are focusing on plant-related bioresources. And this is also where the approach of the ATB research in the area of healthy food started. So we consider in our research aspects of fruits and vegetables, so the composition, the physical properties, microbial diversity of the raw material, as well as of alternative bioresources like macroalgae and insects. And we consider here also the co-cultivation options. That means how to bring these different biomaterials together, starting from the cultivation. Then we go into the processing, where we consider non-thermal processing and gentle processing, and also green extraction techniques to deliver finally minimal processed food, vegetables, and other products. But also tailored packaging is a topic which is of importance for us. Modeling the development of sensors, but also extraction of certain protein lipids or other food components. And to do it properly, we have to know about the material properties and the microbial dynamics along the entire processing line to control, let's say, the product process interactions. And finally, we want to end up with safe and high quality food and food nutrients. Probably we develop new food materials for consumption. We focus on the food loss reduction and the side stream valorization. And with all these approaches, we want to contribute to sustainable and resilient future food systems. So I would like to start because it's also about inspiration, and this is something I would like to mention here, the history of edible insects. So I'm pretty sure that you are familiar with the potential of edible insects, and a lot is known nowadays about this, but this was starting, let's say, 12 years or 13 years ago with the book published by Arnold von Heus from Wageningen. And he published a book at the EFFO for edible insects and human consumption. And this was really boosting the research in this area. And at that time, we already knew that more than 2 billion people worldwide are eating insects from time to time. There are more than 2,000 edible insect species which can be used for human consumption. It was also known that there is a certain benefit because of the high generation cycles and the high feed conversion efficiency of insects. They need less space and water if you rear them, and they emit lower amounts of greenhouse gases. Another interesting part here is that they can be used to valorize organic waste. But this is related, of course, to the legal aspects. And last but not least, they are really rich in nutrients, especially in proteins and lipids. And so there was a huge potential. And to get those insects, of course, you have to think about cultivation or rearing. And there were some interesting studies, even relatively old, let's say 10 years ago or so. And the idea was to make money out of it with these small rearing boxes on the left-hand side for crickets or grasshoppers, on the right-hand side for black soldier flies. And I think it looks fancy and interesting, but I don't know about any device which was sold so far, and it is of course extremely limited, and because of the space you have to provide feed quality, a certain feed quality to get the insects grown, and of course it's also an aesthetical aspect to have this kind of device in the kitchen. On the other hand, there are rearing devices like these ones here shown on the left-hand side, top and bottom. These are simple devices to rear crickets. So you see it's very simple and a lot of manual work. On the right-hand side, you see black soldier fly rearing and the black soldier fly larvae on the bottom. Of course, if you want to be successful, there is a need for industrialization. And this was also realized during the last decade, where a lot of money was spent to get a production or to obtain a production on industrial scale. And this is how it looks like nowadays. So huge facilities for insect production. Right-hand side top, it's in the Netherlands. On the bottom, it's in US. And this is really a huge development in short time. On the other hand, the producers are fighting with, let's say, the other biomaterials coming from plant-related sources like soy, because the costs are much higher for insects when compared to soy, for example. And this is something which challenges these companies. However, if you consider those kind of new bioresources, you have to know how to use these kind of insects in this case as a whole or just a fraction. You have to consider safety aspects. What about the microbial load? What about other contaminations which should be prevented? What about toxins, allergenicity? All these aspects must be considered. And nowadays it is known that we can use at least 4 species for human consumption. So it is a huge step forward integrating this kind of food material into our diets. If we talk about processing, there are different processing pathways, as you can see here. So on the left-hand side, this is related to the whole insect. So let's say producing fresh insects, for example, or dried insects, or frozen insects, or just an insect powder or insect paste. But you can also think about the fractionation. So where you get finally an insect oil, the defatted insect powder, or just fractions like the proteins or the chitin, for example. And if you consider these nutrients, these can be directly implemented into the food production process. So where you enrich food with proteins or lipids coming from insects. And on the other hand, the side streams, so the waste, cannot say waste, let's say the side streams from the food production can go into the rearing so that the insects convert the side streams into say proteins or lipids. And this can be again then used in food production. On the other hand, also in insect rearing, we have some residues, so-called frass. So that means some skin, excrements, whatever. And this can be used also to produce biogas, for example, or fertilizers, which can be used to improve the soil. And with this circular approach, insects can also contribute in this biovalorization process. However, with respect to food, the nutrient quantity and the quality is determined by the production process, let's say the rearing, the cultivation, quantity, and also the processing part. And then with respect to human consumption and healthy aspects and human diet, we have to consider also the nutrient release and the availability in the body. And this is also determined by the food structure. And this is something we will also hear later on in the pitches, where food structure is also considered. Here are some examples of food coming from insect or insect-based food. So very simple snacks where you have roasted insects together with dips or whatever. Then there is some, let's say, tomato sauce with insect, like bolognese sauce or al ragu. Then we have insect meatballs, chips, pasta, protein bars, or just flour. And on the right-hand side, there are some products which I call usually some, let's say, funny products, which probably does not make so much sense just because of sensation. So gin with black ants, or lollipop with black ant, or sea salt with black ants. But when you ever have the chance to test or to taste a roasted black ant, then you should do it, because then you see that it is, or you feel that it is extremely sour. And this is a completely new aspect here, because then it does make sense. Yeah? So having this differences of sweet and sour in the lollipop, or even having sour taste in the salt, this is a completely new aspect with respect to alternative food resources, and this probably will be taken into consideration much more in future. Of course, there are also other biomaterials like macroalgae, has also interesting amino acid spectra, contains a lot of different nice nutrients, and can also be cultivated onshore. And this is a device which we use here at the ATB to cultivate, in this case, a red algae and a green algae. And if you want to do so, then first you have to find the right algae, which can be used for human consumption. You have to isolate it, and then you have to find the best conditions to grow it. And then, since we are in salty water here, You can also consider other biomasses, and this is more or less the newest one we are considering here with our research or within our research. It's a jellyfish, and this is a very special jellyfish, let's say laying on the back, so tentacles up, and this jellyfish is called Cassiopeia andromeda, and can also be used for food consumption, and you can cultivate it also indoor, as you can see here on the pictures. Within a huge national research project, we are considering all these different biomasses at the moment, including halophytes, so salt-tolerant plant materials. And the idea is to think about decentralized production of fresh food materials in growing cities. And this is an ongoing research. And if you're interested in this, I can also give you some more information later on. Within the project, we also try to develop some different food. As you can see here, for example, a halo smoothie, that means a bit salty smoothie from halophytes. We try to have jellyfish crisps, but to be honest, it is not really crisp. It's more chewy. Pesto from halophytes. You can also find palmaria fried and so on. So different food materials coming from these sources. And then we are coming back to food processing, which can be transferred from this insect processing pathways. You can also consider, let's say, more or less similar pathways for other biomaterials like macroalgae or jellyfish. And the different pathways can be supported also by new technologies. And these new technologies are also in parts well known and industrialized already. So we have high pressure, which is applied on industrial scale. We have pulsed electric field also used on industrial scale. UV can be applied. Here on the left-hand side, we have irradiation, ozone, ultrasound. But one technology is not applied, as I know so far. It is cold plasma. And cold plasma is an ionized gas showing antimicrobial activity is at room temperatures. And this offers also a wide range of application in pre-harvest and post-harvest fields in the area of agri-food processing. And here you, for example, can improve the germination of seeds. You can affect the plant growth and, let's say, stress reaction with increased amount of flavonoids. You can inactivate microorganisms. You can change functionality and modify the food properties, or you can also affect packaging film. So wide range of application. But to do so, you need certain devices. And this is a very new picture taken from colleagues from the Leibniz Institute for Plasma Research and Technology. They invented this plasma generator here on the left-hand side. And together with a column washer, you can produce plasma-treated water, also plasma-processed water. And this water can, for example, be used in industrial salad washing line, as shown here. And with the application of plasma-treated water, you can reduce the microbial load of about 2 to 3 log cycle. So it is a really interesting approach. And at the same time, you have to assure that there are no toxic components remaining on the salad leaves. And this was also proven in a recent study. So last but not least, we have to consider, let's say, a holistic approach. And then we are coming to the biorefinery concept, which is presented here. So it is a biomass production. It's a wide range here, starting from forestry, but also algae, edible insects is considered. And on the other hand, we have some biogenic residues. And those biogenic residues can also be obtained from salad washing, for example, if you remove the outer leaves. And this concept is about the material use and the valorization and the optimization of all those processing networks. And this is supported by machine learning procedures, by digital twins. And this concept was also published last year. And I think we have to take this into contribution for the development of entire future food systems. And if you are interested in these kind of research and topics, let's say alternative bioresources, plant-related materials, sea-based, insects, fungi, cultured meat, and so on, then I would like to invite you to contribute to this Congress. It's called Insects Plus. Plus is for all the other biomaterials I mentioned before. And the idea here is to offer a platform to exchange research ideas from cultivation over processing to application, from lab scale to industrial scale. And the first Congress was held in Gothenburg last year. And the next one will be next year in Berlin. And we are organizing this, and last time there were a contribution of about 400 people. And so, yeah, please save the date if you are interested in. And with this nice picture, also taken from the National Project Food for Future, where you also see these alternative bioresources, I would like to thank you very much for the chance to share some of our ideas with you. And I'm looking forward to a fruitful discussion. Thank you very much.
Thank you very much for the very nice and very informative presentation. And thank you also for being exactly punctual with the time. I would like to mention to everyone again that they can write questions also in the chat or in the Q&A, and we can forward to you, and this goes as well for the presenters. They can also write questions during the course of the webinar as well. And I think maybe we have time for one small question. Also, maybe one of the presenters, if they're interested to ask something, it would be a very good opportunity. If not, then we can maybe proceed with the topics. So I will give the word now to my colleague, Dr. Robert Sevenich, and he will proceed with introducing the topics and the speakers. The floor is yours, Robert.
Yeah, thank you, Marius. Yeah, now we come to the core and the heart of this webinar. In the beginning, when we revived this group, we were thinking about what kind of webinar we would like to do, and then we thought, yeah, it would be interesting to hear from the younger people, because a lot of us are now basically today, to be honest, and that's a true fact. It's my 10th anniversary of defending my PhD. So I've been in that game for a long time, and we were very surprised and very happy to see so many submissions for our first webinar. And I can't for some reason move the slides, although I try to take control, but it's still loading. Maybe let me, now I'm going through, here we go. Okay, there's one slide. Okay, well, as said, we try to, based on the different topics we provided to you, like the product formulation, the novel food ingredients, and the food processing strategies. We got so many submissions that we could not implement them all into one webinar, and therefore we selected some where we thought it is fitting. We tried to find a good mixture of beginners, intermediates, and people who are close to finish their PhD. And so without further ado, I would like to present the people who will be presenting in the first topic or first pillar in the product formulation pillar. So basically, we have Esther Stes from the KU Leuven, Chonrong Ping from the China Agricultural University, Nienke van Ittersen, Wageningen University, Joel Armando Nguyen, University of Bologna. I hope I pronounced the name okay and fine. And without further ado, I would now give the floor to Esther. You could just take over the presentation, please. Keep in mind that you have 5 minutes, and after each topic, so after, in that case, those 4 presentations, we will have time for Q&As, and we will collect all the questions that can be either put in the chat or asked directly. Okay, so Esther, I'm giving you the floor. You're the first to start it. Let's get going.
Thank you so much. Good afternoon, everybody. I'm sure you've all already seen articles that say something similar to this on why we should eat more beans and legumes. And this is just one example of an article. There are multiple examples that say why pulses are good for your health, why we should be eating more pulses, why they are good for you, and the list goes on and on and on. And basically, what all of these articles are saying is to eat more beans, please. But why should we eat more beans or pulses in general? Pulses are a very sustainable and a very healthy food source, which is why it is recommended to eat 50 grams of dry pulse seeds every day. This corresponds to about 100 grams of cooked seeds. Now, I don't know about you, but I don't reach that goal, and I'm not the only one. In Europe, the current daily consumption of pulses is less than 10 grams, so we see that there's a big difference between the current consumption and the recommended consumption. Therefore, in my PhD, I'm trying to increase pulse consumption by making pulse-based flours. My name is Esther, and at the Laboratory of Food Technology at KU Leuven, Belgium, I'm trying to incorporate pulse-based flours into products which we already eat more on a daily basis so that we can increase pulse consumption in a very easy way. I already hear you thinking, Esther, such products already exist, and this is true. In a supermarket, we can, for example, find lentil-based pastas. But such products are made with what we call traditional open cell flour. What I mean by this is that to produce such flours, we have to mill a raw pulse seed, and intact cells that are present in a pulse seed, they break open. So we lose the cellular integrity present in a pulse seed, and what we get is actually a flour in which all the nutrients, such as starch and protein, are free, as we can see here on the microscopic picture. However, if we lose a cellular integrity, we actually also lose some of the health benefits of a pulse seed. But pulse seeds have a very special property that we can make innovative cellular flours out of them. If we, for example, cook a pulse seed, so just simple cooking as you can do at home, and then blend them or mix them, the cells will not break open, but they will actually slide off each other. We call this cell separation, and what we get is a flour in which intact cells are present. And in these intact cells, nutrients such as starch and protein are bioencapsulated by the cell wall. In my PhD, I'm mostly focusing on how the microstructure of these flours impacts their digestion properties and thus their potential health properties. If you, for example, take a look at star digestion, we do this in the lab, so this is not in real humans, but we can clearly see that this purple curve, which depicts the cellular flour, is digested much slower than the open cell flour in blue. This is because in the cellular flour, digestive enzymes need to overcome multiple physical barriers to get to the substrate. So they have to overcome, for example, or diffuse through the intact cell wall, which is why starch is digested much more slowly as compared to the open cell flour, in which starch is readily accessible for the enzymes. Now, such a decrease in starch digestion rate has been linked to multiple health benefits, such as a prolonged feeling of satiety and a lower glycemic index. In this sense, a cellular flour could thus potentially be more healthy than an open cell flour. This is not only the case for pure cellular flours. If we blend both flour types together, we can also see that from very low proportions of cellular flour on, so for example, this orange curve only contains 10% of cellular flour, we already see that starch digestion is slowed down and thus potentially will have health benefits in this blend. This, of course, shows that with very, very simple processing steps that you can even apply at home, we can largely impact the microstructure of our ingredients with the potential to affect the digestion kinetics. And this brings about perspectives for the use of such innovative cellular flowers, or even only low proportions of them, in products with impacted glycemic responses and thus potential health benefits. And this is, of course, what I'm working on right now in my PhD. I'm trying to incorporate such flowers into food products. So in the beginning, I showed you an article that said to eat more beans, please. But maybe in the future, we can also eat more products with cellular bean flour, please. Thank you for listening.
Yeah, thank you, Esther, for being perfectly on time. So we can move to our next speaker, Chunwan. You could take over the presentation mode, please, and then you can get started. As mentioned before, questions will be done after all the speakers within topic 1 have presented.
Thank you. Can you see me? Can you hear me?
Yep.
Okay. Hi, everyone. Good afternoon. I'm Chen Yanping from China Agricultural University, major in flavor chemistry. I'm very honored to be invited by the EFFoST. Today, I'm going to talk about how tangerine peel affects the flavor and hydroxylic aromatic amines formation in deep fried rabbit meat. So rabbit meat is globally consumed due to its rich source of nutrients but low fat and cholesterol. Tangerine peel is a Chinese typical medicine. It's rich in flavonoid, phenolic compounds. The medical prosperity change with ripening years, like 5 years, 10 years, 15 years, and their prices increase with ripening age. So today, by combining these two ingredients, a very distinctive and flavorful traditional Chinese dish can be prepared. Seems very tasty, right? However, this dish requires deep frying at 240 degree, which generates desirable flavors, but may also promote the formation of HAS, which is high hydroxylic aromatic amines. So, but they are also associated with potential health risks. Due to the tangerine peel contains antioxidant compounds, we hypothesized that it could reduce HA formation during the frying process. So let's start with question A. How does peel ripening year affect the aroma profile? So we detected 35 compounds overall. Flavor compounds diversity was highest in 3-year TP and lowest in 15-year TP. Notably, major alkane, talpene, especially D-limonene and gamma-talpene, decreased with aging, possibly due to the time and temperature during the ripening process can affect the rate of oxidation or lipid degradation, thereby decrease the concentration of flavor compounds. And the second question, how does TP affect amino acid in the fried rabbit meat? Proline was the most abundant amino acid, and the 15-year peel showed the lowest concentrations, while 5-year peel had the highest total free amino acid content overall. And figure B shows that after eating TP, the total free amino acids in the fried rabbit meat decreased. And the 15-year TP caused the strongest decrease, with key amino acids proline, aspartic acids, alanine, and serine. So figure 3A is the most important one, shows 5 HAAS detected in fried rabbit meat, and 7-DIMAQX was the predominant one. As we expected, adding tangerine peel significantly reduced HA formation. However, interesting, the inhibitory effect was not positively correlated with aging time, because for Chinese 15-year TP is really useful for us, our health, at least our concept. But for 3-year TP, 84 or 6% completely inhibited MEIQ and MEIQX, but that's not enough. The best overall result was observed with 5-year TP at 4%, which reduced 4 type of HAS by about 95%. 10 and 15-year TP showed strong suppression mainly at 2%, reach up to around 94% for 15-year TP. So we speculate that 5-year TP may provide more bioavailable phenolic compounds, while prolonged aging could reduce some reactive bioactives. But from a practical perspective, this is actually good news because 5-year TP is much more affordable than 15-year TP, even much more cheaper. Yet it still shows excellent inhibitory performance. But due to the time constraints, I wasn't able to cover everything today. I'd be happy to discuss it further if you are interested. Finally, thank you for your attention. I've been focusing on flavor change in prepared dish, and I'm grateful to EFFoST for this opportunity, and it's been a pleasure meeting so many excellent researchers here. Thank you very much. Thank you.
Yeah, also thank you to you for staying perfectly on time. So without further ado, I would now to introduce to you Nyenke van Ittersen for the next talk. Could you please take over the presentation and then start?
Yes, thank you so much. I think it works like this. So my name is Nienke, and today I will talk to you about my PhD project called Snack Petition, where we're on the search for texture and flavor perception of snack bars. I thought to introduce myself a little bit, as there are so many of you here. So I myself have a bachelor's and master's degree in food technology at Wageningen University and Research, and I started my PhD around 2 years ago. And during my PhD, I'm affiliated with 2 different chair groups, the Food Quality and Design Chair Group of Vincenzo, and also the Human Nutrition and Health Group of Sensory Science and Eating Behavior. And you'll see that my topic comprises both. So regarding the knowledge gap, there is a trend of snackification going on, where we shift from 3 meals a day to more frequent snacking. And this demands for more healthy and more natural snacks, but obviously they have to be tasty as well. And that's why snack bars had an upcoming market. And you can see here an example. We also know that the market size is expected to increase even more the upcoming years, making it a relevant category for product developers to focus on. But we also know from sensory science that texture and flavor are still the most important attributes contributing to food choice. And these snack bars, as they are very heterogeneous, they consist of many different parts. They allow us to play with many different parameters, such as mechanical properties, for example, the hardness. We can play with the geometrical properties, such as the shape or the particle size. We can also play with the flavor. So where do you distribute the flavor in the matrix? And all these knowledge gaps together kind of brings me to the aim of my project, which is to investigate the impact of food structure on sensory perception or processing, flavor release, and eating behavior of heterogeneous snack bars, which is very multidisciplinary. And I'll guide you through what I've been doing in my PhD and what I'm still planning to do. So in the first study, we had around 200 participants evaluating 30 commercial snack bars. And part of the participants evaluated this with sensory attributes, and the others evaluated how much they liked the bars, how healthy they thought they were, and how natural they thought they were. And by analyzing this, we found out some drivers of liking and healthiness, which can give some guidelines for product developers. Results will hopefully be published soon. For the second study, we had around 40 participants that evaluated a subset of the bars from the first study, and we cut them in the same geometry to kind of isolate the effect of texture. And we also were curious to see whether flavor might have an effect on these parameters as well. So we had 1 bar in 3 different flavors as well, while keeping the texture constant. Then again, we took a subset of 3 of these bars, a bit extremes, and we also cut them in different geometries, where we varied the length to width ratio, and the weight within each type of bar was the same. So we were really isolating the geometry effect here. The first part of my PhD is focused on commercial bars to really get a feeling of what is already on the market. But at the moment, we're also moving more towards making our own bars. And the first study we did on that was with around 40 participants, and we varied the nut particle size by blending a commercial bar. And we also added chocolate to give a more lubricating effect to see whether that has an effect on all these parameters. And for the future, we still want to do a study on flavor. So we focused a lot on texture, but flavor plays a very important role in your decision making. And also when you want to take another bite. So if something is very lingering, you might wait a little bit taking your next bite. And then we're planning to do a final study where we can combine all these guidelines to give guidelines to product developers to design snack bars that promote a healthy eating behavior. That's kind of the goal. And then the first and the second and third study have already been performed. And for the future, we're still focusing on this part. And then the methodologies, as it's very multidisciplinary, I thought to focus on this. So we use static sensory perception, which basically means participants have to rate on a line scale their attributes. We also use dynamic sensory perception, which means that it's throughout the time. So throughout consumption, we ask them to either check attributes or follow a certain attribute and indicate the intensity of that attribute. For us as researchers, this looks like this. It's preparation of a lot of different samples for participants to eat. And for participants, the setting looked like this. So they had a camera that was filming their eating behavior, so their chewing behavior. And they had a bar that was on top of a hidden balance. So they were not aware that their bite size was being measured, but very useful information for us to know. And this is a lot of video annotation to get the eating rate and the number of chews out. And then we also have some subjective measures such as self-reported appetite. So how hungry are you or how full do you feel right now? As well as some consumer perceptions where we try to link this liking and healthiness together. So that's my PhD project. And if you have any questions, feel free to ask later.
Yeah, thank you so much. And also thank you to you for being on time. So now we move to the last speaker within the topic 1 product formulation. Joelle, could you please take over the presentation and proceed? Thank you.
Oh, sorry. Okay, can you hear me?
Yeah, we can hear and see you.
Okay, thank you. Okay, good afternoon, everyone. Thank you very much for the opportunity to present my PhD project, which is an industrial PhD in collaboration with a consortium, a public consortium specialized in the production of corrugated cardboard for the packaging, let's say, of fruit and vegetable. Okay, before starting, I wanted to present you some data already mentioned by Professor Oliver Schotter related to the food waste and food loss and waste, let's say, in the world. As we already know, 1/3 of the production of food produce in the world is loss or waste, so never reach the fork of the consumer. And among this, let's say almost half is represented by fruit and vegetable. This has a really, let's say, notable consequence on the economical level, at the environmental level, and even at the social level. This is why the United Nations in the agenda, let's say 2030, integrated this issue. So the objective at 2012, 1.3 was to, is to reduce by 50% this loss and waste before 2030. So talking about, let's say, fruit and vegetable, this is a part of my PhD. We know that, let's say, fruit and vegetable are really perishable products related to their metabolic activity, but also due to microorganisms, because they are really susceptible to the colonization by microorganisms, especially mold, which can also produce some toxin. So this can cause not only problems related to the loss, but also food safety issues, let's say. So it was urgent, it's really important here to find solution in order to tackle this problem, this issue. So this is the main objective of my PhD, so to extend the shelf life of fruit and vegetable, let's say in general. So to achieve this, let's say a packaging can play a really important role in this sense. So that's why we decided, we say, to find for a solution that is able to, let's say, be sustainable also and reduce the use of plastic, for example. So one biomaterial in this case that has emerged in the last day as really sustainable and fit this requirement is represented by bacterial cellulose. This is one of a similar product. It's similar to plant cellulose. Just to mention that cellulose is one of the most abundant polymer on the planet. So it's a renewable material, it's biodegradable, and for some application, it can even represent a good alternative to plastic. This is because it's highly stable, thermally stable, let's say, and has good mechanical property as well, and is completely biodegradable. But in our case, one of the, let's say, feature of bacterial cellulose is its porosity, and the amount of hydroxyl group that it contains, let's say, which can be exploited to incorporate some let's say, compounds, some active compound, antimicrobial compound or antioxidants. In this case, we can produce a kind of so-called active packaging. So to realize this, we started from scratch. From raw material like kombucha, we isolated microorganisms able to produce cellulose, in this case, acetic acid bacteria. We characterize them, we screen them, and then we selected the most performance. In the next phase, we choose agro-food byproducts, in this case, to have a kind of a circular economy, in this case, circular approach, in this case, to produce cellulose. We use beet molasses and cheese whey. Then we optimize the production process, and we choose natural antimicrobial deriving from essential oil. In this case, we incorporated them into the bacterial cellulose produced and obtain a functionalized film, which will then coat it with the cardboard, and then the shelf life evaluation, the effect on the fruit and vegetables. So we expected with this approach to improve the quality and the safety of food product, also to save energy and money, because the product can be also, let's say, stored at higher temperature. And this can be suitable because it respects the sustainability, environmental sustainability. But the most challenges, let's say, the most challenge at the moment, as even Professor Schulte mentioned before, is the industrialization. From the transition from the lab scale to the industrial level. So thank you very much for your attention.
Yeah, thank you to all the first speakers within this topic for being perfectly on time. We have some time for questions. There are currently no questions within the chat, but I have several questions written down actually for all four of you. Maybe we start with the first presentation from Esther, and maybe she can give some. Oh, somebody raised her hand. Okay, somebody raised his hand. Or write the question in the chat. I think that's easier, and then I will ask the question. But I will start with Esther. Is that cellular flour you described something specific of beans, or are there also other let's say wheat or hulled vegetables where this would make sense to look into?
This is a very legume-specific property, let's say. So in cereal-based foods, this is not possible because of different compositions in their cell wall. So we need pectin in the cell wall to be able to get this cell separation and to get this cellular flour, whereas in wheat, this is not the case.
Okay. Okay, thank you. And I have one question to Joelle. Do you think that the sorry, there's a lot of going on here. Do you think that the bacterial cellulose could replace synthetic polymers at a big scale, and will it be affordable?
Oh, thank you very much for the question. As I pointed out at the end, is the big challenge at the moment. I mean, since all is almost related, let's say, is more to bacterials, so to microorganisms, it's a little bit difficult to industrialize the process, let's say. But there is still a possibility, let's say, to optimize the process to reduce it, because yes, for sure, the cost is really important. It's an important aspect to take into consideration. But we are trying to reduce it. That's why we decided to use, for example, agri-food byproduct. But there is still many things to do. There's still work to do in order to be affordable, to be challenge product, let's say, to challenge plastic, for example. But I would say that in many other fields, like in biomedicine, in cosmetics, is something that works very well due to its characteristic, I think.
Yep, thank you for your answer. That made total sense. And for Nienke, it's also some question. First of all, so this will be like a package of questions now. First of all, what do you consider healthy in terms of snacks? Secondly, why not change the eating patterns that are currently existing and building something new? And also, aren't you supposed not to snack too much because this is also not too healthy for your teeth?
Sorry, if you can go back to the first question.
So the first question was, what do you consider healthy in terms of the snacks? So what kind of ingredients, like the percentage of the different ingredients, would be interesting here. And then I know it's kind of a mean question to ask, but I find it interesting, or I would like to hear your answer on this one. Why not? Why shouldn't we change the current eating patterns we have instead of these something that goes into something that's actually making the needs available? And what was the other question I had? The snacking is not healthy for your teeth.
Okay, so to answer the first question, in the first paper, we considered healthiness based on the Nutri-Score. So this is, I think, one of the most renowned measures we used. And we do see from literature, there are a lot of studies done on what Nutri-Scores these kind of products have. And the majority has a bad Nutri-Score. So then we say like D and E. Only a small amount, like 15%, has a good Nutri-Score of A to C, let's say. So for the first paper, we kind of looked at, okay, are products with a good Nutri-Score also perceived as healthy? We found actually that this correlation is not so straightforward. So this is also something to look at for product developers. And then your second question regarding the eating pattern, I think we do see that, of course, there are different ways to solve these problems, but I do think that it could help a lot if it's done in a way that consumers are not necessarily aware of it. So there are many strategies for designing products, such as making a hard texture where you're forced to eat slower, which gives you a longer satiation, instead of trying to change people to stop snacking. I think we should try to focus on making the snacks more efficient, more healthy, more satiating, because I do think snacks are there for a reason, and people are consuming there for a reason due to being in a hurry, not having time to cook. We're in a fast-paced lifestyle in the current century. So I do think it's better to maybe focus on the products than on really changing behavior here. But I think we can help a lot as product developers. Yeah, and then regarding the teeth, obviously, we realized also that, for example, an attribute like hardness affects your eating behavior. But of course, there's an optimum there. If something is too hard, this will also not be pleasant to eat. So yeah, we're not measuring anything regarding like how much is stuck to the teeth, like somebody mentioned in the questions. But yeah, we are realizing that every attribute has their optimum, so that we have to keep that into account as well in these design guidelines.
Okay, perfect. Are there any other questions? So here's another one to Esther maybe. Depending on the treatment, what is the impact of oxidation of the substrate and how to prevent oxidation of the flour?
Well, I must say, my PhD specifically focused on the digestive properties, but it's part of a bigger project. And in a second PhD, we're also looking at like sensory properties of these two flour types. And I know that when we make a cellular flour, we have to apply a thermal treatment at first. And then we have, in terms of sensory properties, much more Maillard-related reactions. Whereas if we simply mill the seeds or the raw seeds, we get more enzymatic lipid breakdown and more of the grassy or beany flavor or smell normally related to these pulses. So in that way, yeah, these oxidation or these reactions can really impact the sensory properties of the flowers. But I do have to be honest that that's not my expertise right now. That's someone else doing that.
Okay, perfect. I think we have one more question I would like to ask Chunyuan. And the question is there a potential explained mechanism how TP affect the amino acid in the fried rabbit?
Yeah, that's a great question. Before I researched some article that I think it's because the tender peel, inside they contains a lot of antioxidant materials compounds, and they also have the amino acids, and in the fried rabbit meat they also have something, and when they together frying, because this dish have to fry and stir fry and stew. So I think in this process they were decreased, they're changing, but the specific mechanism we still have to do more experiments to explore it. So we still have to explore it.
Okay, yeah, thank you for this answer, and thanks again for all 4 of you for presenting in this topic 1. And now we move on to topic 2, novel in brackets food ingredients. I think somebody it's coming back up again, I think. Yeah. Okay, so within topic number 2, perfect, thank you. Topic 2, novel food ingredients. We will have 2 talks, one from Zourav Nepal from the ATB and Lucas Botso from the Wageningen University. We have the same rules and same guidelines as for topic number 1. So therefore, Zourav, you can take over the presentation and please enlighten us.
Are you listening me?
Yeah, we can hear you.
Yeah. Okay. Thank you so much for having me here. And I am Saurabh Nepal, currently working as a scientist in EFFoST. And moreover, I am also a doctoral researcher at the University of Göttingen. Today, I am going to present you on the topic from hope biomass waste to fiber, a sustainable utilization strategy. Here today I'm going to talk about a plant which name is Humulus lupulus and commonly known as hop. So this hop plant, uh, some of you may know that this hop plant is basically used for harvesting of the cones, and which cones are used for brewing purpose, and the cones provide aroma and flavor to the beer. And after the cones are harvested, the biomass is chopped into small pieces and discarded. So today I'm focusing on this discarded part. So this biomass is discarded and ends up with ends up in the soil with minimum utilization. And this biomass, biomass being the bast fiber plant, it has a great potential. Here in the figure you can see the typical bast fiber stem of this plant, and this bast fiber plant is used to produce the bast fibers. So having this potential, my PhD project focuses on the proper uh, storage technique and processing techniques. And there are few challenges for proper utilization of this biomass. The few challenges are: this biomass is harvested once a year, and during September, and large amount is produced within a certain time. And here you can see around 100,000 tons of this biomass is produced in Germany per year. And there is lack of proper research on storage technique and appropriate processing technique. And my research questions are: how storage duration affect the chemical composition of this biomass, and how storage duration affects the particle size distribution of the process output. So for the method, like I'm focused on storage method and processing method. And for storage method we choose insulation. As we know that insulation is well established method for the fodder production. So here we inside the biomass and store it for various storage duration. After certain storage duration we do the processing, and for the processing we use twin screw extruder. In this twin screw extruder we pass the feed the biomass, and the biomass goes to the screws and defibrated and comes at output. And after the defibration we usually dry the biomass, and if needed we do milling to break up the agglomerates. So here are the results of the storage. Here during the storage of the biomass we look at the storage duration of 2, 4, 6 months and 12 months, and we can see that the structural carbohydrate like cellulose, hemicellulose, and the lignin content, they are basically stable during the storage duration. So here we can conclude that the insulation can be used for proper preservation of the biomass. And after processing we can get the fibers such as in this picture, and with the processing at different storage duration. So if we go for longer storage duration we get the finer materials, and at the end we can see the use of those fibers in as a material. Here you can see the pipe, and here you can see the frame of the drones. So here is my conclusion. The conclusion is like structural carbohydrate and lignin are preserved, and extrusion can be robust and technique for production of the for the intermediate, and extruder grade fiber can be used for making lightweight composites and so on. Thank you so much for your attention.
Thank you, Zura, for being on time as well. So we will move directly to the next speaker, to Lucas from Wageningen University, who I think already took
I
over.
already have control.
Yeah,
Yes.
you already have control. So without further ado, please go ahead.
Perfect. Thank you very much. So good afternoon, everyone. My name's Lucas, and I'm going to be telling you a little bit about what I'm doing as my PhD. And I'm actually very happy with Oliver's presentation because he saved me a lot of the work with my introduction. So my research actually focuses on how we can convert microbial cells into food ingredients. More specifically, I work on yeast, bacteria, and microalgae, and these are often defined as microbial biomass. My research particularly focuses on the conversion then of these cells into food ingredients. So after the bioreactor, here's on the left actually is a picture of the individual cells, and on the right is a picture of how the product looks right after the bioreactor. You can actually already find some of these products in the supermarket, and then they look somewhat like this. So this is the chlorella powder. It's a microalgae. And as you can see, apart from the price, which is very expensive, you can see that it has quite a nice nutrient composition. It has 58% egg white, is the literal translation of protein from Dutch, but quite rich in protein. The problem is, however, that not a lot of people will end up consuming this powder as such. So we are lacking nowadays knowledge on how can we process these ingredients into food, these cells into food ingredients, and then how can we then use these ingredients to make food products that people will actually taste. And these are actually the two main objectives of my research. In order to make these ingredients, my research starts from two premises: that we need to break the cells open, because that's nowadays often very often not done. And then we actually afterwards we might need to purify different fractions out of this. With this aim, I want in my research to use a process that allows me to process different kinds of microorganisms, and I do this with bead milling and centrifugation. And bead milling as a process to break the cells, which is a mechanical process that is non-cell specific. And afterwards, I use a simple process such as centrifugation to separate the biomasses into soluble and insoluble extracts. However, before we actually dove into depth into, well, what do we get out of these ingredients, we actually needed to know about the composition. So what is in the cells? And this is very important information for us to track the performance of the process, and when we have the ingredients, to also to assess their possible application. And also outside of the scope for my PhD, this is also very important information for the novel food dossiers. However, microbial cells, they are not your average food product. So they have very complex cell structures. So they have a cell wall which is very rich in hard to break polysaccharides, cell membrane with both polar and apolar lipids, and a cytosol with containing RNA, amino acids, which are forms of non-protein nitrogen. Therefore, my first objective of the PhD was to look at how can we accurately assess the composition of these microbial cells, and hopefully these results will follow up soon. But the ultimate goal of my research is to actually study the link between different processing conditions, different grades of refinement, and the technofunctionality of these cells, such as the gelling properties, foaming, and emulsifying properties. So to sum up, I hopefully have been able to give you an insight into why microbial biomass can be a potential new source of food ingredients, and that knowledge on the extraction and application of these novel food ingredients is very limited, and that accurate compositional data is very important and lacking to both provide insights into their potential application, but to also to understand the processes. So thank you to you listening, also for my collaborators, and looking forward to your questions.
Yeah, thank you to you as well, Lucas, for being and staying on time. So I've a couple of questions. I think we will start with Zorav. Is there an option to manipulate the structure of the carbohydrates to affect the functionality or the physical properties? Have you looked into this?
So right now, like we haven't looked into it. Like right now, what we are doing, we are basically in the very initial phase. So we just produce a fiber and incorporate with the matrix and make a hope fiber reinforced composite from that matrix. So we right now, no, we are not looking to change the structure of the carbohydrates or something like that.
Okay. Do you have any application potentials of the composites? I saw a picture there, but I wasn't really sure what kind of application it is? Like, is it for, yeah, do you have anything in mind?
Yeah, there are two pictures. Like one is the frame of the drone. So like a drone used is the frame. So like we can use this hook fiber reinforced composite for making the frame of the drone. And second one is the pipe. So we can make the pipe out of wood these composites. And yeah, here we can also make the decking material and other material and some parts of the automobiles also. So yeah, we have wide range of use. So yeah, we can say the hoop fiber is the alternative to the wood fiber. So yeah.
Okay,
So
thank you.
Yeah.
Yeah, due to time, I will also give Lucas the chance to answer to some of the questions I received here. The first question is, why is it important to break the cell membrane / walls to produce food prototypes? And then also, could you elaborate how the bead mill to open the cells, how you can scale that up? Is this a possibility, or are you thinking maybe about using other, let's say, more gentle technologies like pulse electric fields? Could you give us an idea about how do you think like scaling up, and why is it important to crack them open?
Yes, so we start from the hypothesis that actually these cells, they have very tough cell walls in most of the cases, and as long as the cell is not broken and you incorporate the cell as such into a food product, all the components which could potentially have a functionality, so let's say a gelling or a foaming property, they will be entrapped within this, well, they will be, say, encapsulated. And that's why we think that disrupting the cell wall is important. And then regarding the choice for bead milling, so in our case, because we are using such a big group of microorganisms, so I actually have 10 different microorganisms in my experimental design, we wanted to look for a technique that was non-specifical. So that's why we went for the mechanical cell disruption. And when you look at technologies which are proven at scale, you very often encounter yourself with either high pressure homogenization or bead milling. And we chose bead milling not because it's the best, it's the one that actually was allowed us the most efficient disruption. And as a curiosity, there's actually some companies already using this technique at scale to produce food ingredients from microbial cells. So it is possible.
Yeah, no, makes total sense. I'm just asking because I'm coming from a department where you do a lot of opening of cells with pulse electric fields. So I was wondering if that was an idea. And I get the point that you have diversity of cells, although there is, I guess, a field strength that will work for all of them. But anyways, that can be discussed at another time. So now we move on to, first of all, thank you for the speakers within topic number 2, novel food ingredients. And lastly, now we move to topic 3, which is called food processing strategies. And there we have 2 presentations, one from Olivia and one from Patrick, both from Wrocław University of Economics. And if Olivia, who's going first in that topic 3, could take over the presentation mode, and please get started.
Okay. Can you hear me?
Yes.
Okay, and you see my presentation?
That we do as well.
Okay. So, hello, my name is Olivia. Thank you for the opportunity to present my PhD work. The title of my presentation is Agarose as an original organoisolator, an innovative approach to structuring vegetable oils and integrating them with proteins. Let me briefly introduce to the background and objectivities of my research. High intake of saturated and trans fats is a health concern, so there is a need for better fats alternatives. The aim of my research is to develop structured lipid system based on vegetable oils using agarose and to evaluate their potential food application.
Now, let me show the preparation process of the hydroleogels. On the left side, you can see the schematic diagram of the preparation method. On the right side, there are photos of the final gel samples. At the top are gels with pumpkin seed oil, in the middle with hemp oil, and at the bottom with rapeseed oil. This shows that stable gel structures were successfully obtained for all test oils. Next, I will briefly present the characterization of the hydroleogels. I evaluated their rheological and textural properties to understand their mechanical behavior. I also measured oil binding capacity, color parameters, and oxidative stability. In addition, lipid quality indices, fatty acid profile, and volatile compounds were analyzed. Storage stability and statistical optimization using response surface methodology were also performed. Finally, the hydroleogels were tested in a model food project, which was cookies. Overall, the gels were stable but relatively very firm, and is dependent on the agarose concentration. They showed more breathness than elastically, so the spreadability still needed improvement. Importantly, the hydrogel matrix provides protection against lipid oxidation. Moving to the next step of the study, by applying freeze drying, I obtained oleogels, which are water-free structure where all drops are dropped in the agarose network. Both structures fat were then used as butter replacers in shortbread cookies. The results showed that they performed well in this application. However, despite the promising results, some limitations were observed. Hydrogels showed an elastic but brittle structure and tended to crack under deformation, which makes them unsuitable for spreadable fat application. In the case of oleogels, some oil loss was observed, along with the formation of a dry and crumbly material. Additionally, after rehomogenization, the structure became not uniform. Therefore, future optimization of this system is still required. As the next research step, I plan to improve the structure and spreadability of the system. For this purpose, I will develop ternary system based on agarose, oil, and pea protein. Pea protein is a promising ingredient because it has good emulsifying properties and it's plant-based. I expect that its incorporation will help strengthen the network and improve the functional properties of the gel. And more information about this topic you can find in our review article shown on this slide. And thank you for your attention. If you have any question or would like to discuss this work, please feel free to contact me. Thank you.
Thank you, Oliver, also for staying on time and moving from Olivia now to Patrick, who will be the last presenter. And then afterwards, there will be also time for questions. And if we still have time after that, there can also be some questions to the other speakers today. Patrick, did you take control of the presentation yet?
Yes, I did take control.
Okay.
Can you hear me? Can you see me? And can you see my presentation?
We can see your presentation. Great. We can hear and we can see you as well. So
great.
The stage.
So officially, hello everyone, and thank you for the opportunity to talk to you today and show some part of my researches. My name is Patrick, and I'm a PhD student at University of Economic and Business in Wrocław. My doctoral research focuses on developing a bioraffinery concept for winery waste, particularly grape pomace, generated by the Polish wine industry. At first, why should anyone ever even care about winery waste? At first, Around 75 million tons of grapes are produced worldwide each year, and approximately half of this production is used by the wine industry. About 1/3 of the mass of grapes proceeds for winemaking remains as waste. That gives us about 9 million tons of waste products each year. What's next? Although grape pomace is often used as animal feed or for bioenergy production, its rich chemical composition makes it a valuable source of recoverable compounds. Among them, tannins are especially important due to their high abundance and wide range potential application. When we talk about tannins, tannins are widely known for their antioxidant activity. However, they are also highly reactive polyphenols capable of interaction strongly with biomolecules. Due to their multiple phenolic hydroxyl groups, tannins can effectively cross-link proteins and other biopolymers. Despite their high value, the recovery of tannins still relies mainly on conventional solvent extraction methods. And these processes typically require large volume of organic solvents such as ethanol, acetone, or methanol, which raises environmental and safety concerns. In addition, they often involve long extraction times and high energy consumption. That makes them difficult to scale sustainably. Therefore, there is a growing need for more efficient and environmentally friendly extraction technologies. To demonstrate the potential of pressurized liquid extraction, because example of this green methods is, for example, PLE, so pressurized liquid extraction. I perform comparative experiments using grape pomace as a raw material. Both extraction methods use the same solvent system, 50% ethanol water mixture, allowing a direct comparison of the process efficiency. The results clearly show that PLE significantly enhanced extraction performance. Total polyphenol concept increased from below 40 milligrams gallic acid equivalent per gram of dry matter in conventional extraction to around 60 milligrams per gram after PLA. Even more importantly, the proportion of tannins determined using a protein precipitation assay with a BSA increased from about 25% to about 40% of total polyphenols. My research focused especially on tannins because they represent the most functionally relevant fraction of polyphenols. Due to their strong protein binding ability, tannins can act as a natural cross-linking agents in the next stage of my work. These tannin-rich extracts will be used for cross-linking natural proteins such as gelatin with the aim of developing biodegradable, edible film, and sustainable packaging materials. The experiments were conducted under different extraction conditions, including variation in solid-liquid ratio and pressure levels in the PLE, to evaluate the influence on extraction efficiency. And overall, this study demonstrates that pressurized liquid extraction can significantly improve the recovery of functional tannins from winery waste, as supporting its valorization as sustainable source of bio-based cross-linkers within a circular economy framework. Literature, and thank you very much for your attention, and I look forward for the discussion.
Yeah, dear Olivia and also Patrick, thank you for being on time. Although we are now actually on time. So we have, I think, one question each, also for the two presenting in topic 3. So the first one goes to Olivia. So there are already some hydrogel-based solutions out there. What stands out with the solution you are trying to put out there?
Sorry, can you repeat?
So there are already some hydrogel-based solutions out there. So in comparison to those, what stands out with your research, or what's different?
Difference is agarose because I never use researchers use agarose as hydro organogelator in oleosols because it's not easy. But I use it because it's a natural plant-based polymer delivery from algae, and it's correlated with nutritional trends.
Okay, thank you. And then there's also one for Patrick. Did you think about fractionization and biorefinery concept where you could also recover fibers and maybe some other valuable compounds on top of the tannins?
Yes, the idea is to make concept and at first step recover the tannins and then use the rest of the material as a source of cellulose lignin, for example, and some polysaccharides to convert them to furfural or from lignins to, for example, active carbon. But that's very far from me today because I'm just starting, and that's a plan for future.
Yeah, thank you so much to all the presenters. Well done. I know it's really hard to stay on time to put like your current research on the spotlight and also like pitch it in like 5 minutes. Well done. Great job. So I have one more question. Or Marius, do you want you collected some of the questions, I think, also to Oliver. Is there a specific one we should ask? You're
Sorry, I
unmuted.
forgot to open. Yeah, sorry. There are a couple of them on the Q&A I can ask. So, how many years will it take to have the transition towards insect protein? And also, when using organic waste or side stream as substrate for insects, I guess, is it necessary to apply any pre-treatments to ensure chemical and microbiological safety?
Yeah, so thank you very much for the questions. I think with respect to the insect protein, it is already used and developed so far. So we already have the insects used for human consumption. And as far as I know, also the processing steps are available and can be applied. I'm not sure about the products in which just the proteins are used, but it is possible in general. So that was the first question, I guess. The other one was also related to protein. I think it was biorefinery concept. Was it, Marius?
Oh, it was about when using organic waste, if
Yeah.
it's necessary to pre-treat it, let's say, to ensure chemical and microbiological safety.
Yeah. There are some rules depending on the use of the insects, especially for feed. There it depends also so the question is which side stream you can use. There are some which can be used if it is not really waste, because per declaration, if it is named waste, then it is not allowed to use it. as far as I remember right. And in case of a side stream, it can be used if it is allowed by the local authority. So this is the case in Germany in parts. But then often, as far as I know, you have to have heat treatment, at least 70 degrees C for 1 hour. If you want to further use it, especially for feeding purposes. So for human consumption, I think it's completely excluded to use side streams. There are different legislation you have to consider. But to be honest, I'm not in this direction of the legal aspects, but there are solutions, and it is used anyway. So yeah. Well, Marius, maybe you can also add if you have an idea or if you are more deeper in this
I think
application.
no, I think your answer is actually quite on point. So there's really nothing more to add. It depends on how you want to handle the final product. You have to respect legislation. I think you covered everything. So I would like to thank all the speakers and the keynote speaker for their presentations. I would like to thank all the participants for their participation. If you would like to know more about the working groups and the working group activities, you can also go to the website of EFFoST and you will find information there. And then before we close the webinar, I would like also to invite everyone to the next webinar of our working group webinar series. This will be organized by the EFFoST Working Group Sustainable Food Systems. And the topic will be food safety versus sustainability. Is it two sides of the same coin? So this is an interesting topic and a very interesting question to be raised and hopefully addressed. More information will be announced soon. So yes, please go ahead and you will be informed, and go ahead and find also the working group activities on the website. So I would like to thank everybody for their attention, and yes, so thank you for the successful webinar, and well, until the next one.